glycogen storage disorders, which will be followed by HMP pathway. So before I begin, let me once again quickly reintroduce you to the core features of Oracle. We have the live classes, recorded classes, notes, MCQs, all based on the latest curriculum. Apart from that, we also have personalized counseling sessions to make your medical school journey more enjoyable. So if you have any other needs apart from the academic needs, you can book a slot with us. We have three different subscription plans starting from 15K going up to 50K depending upon your needs. So you can avail any of it, basic, standard or premium. And we have different services available under each plans. For an extra 20% off, you can use my promo code DRBEENISH20 as displayed on the screen. Okay, moving ahead, let's start with today's topic, that is glycogen storage diseases. Before we begin, let me take this clinical case scenario. It says a 6-year-old male child is brought to the pediatrician with complaints of recurrent episodes of hypoglycemia. Hypoglycemia, it says, fatigue, and hepatomegaly. The child's growth and development appear normal, but the parents express concern about the frequent episodes of weakness. Laboratory tests reveal hypoglycemia, elevated liver enzymes, and an enlarged liver. Further investigations indicate a deficiency in glucose 6-phosphatase. Based on the clinical presentation and test results, the pediatrician suspects a glycogen storage disease. What type of glycogen storage disease is most likely affecting this child? Okay, so although the question is very lengthy and it might intimidate you, but after we've done the topic, you'll realize that the question is actually very easy. Sometimes the extra information is given only to confuse you. So don't get intimidated by the length of the question. Read it thoroughly, and as and when you read, a pro tip is that you underline the keywords. Okay. So moving ahead, keeping this clinical case scenario in mind, let's begin with our topic. So introduction about briefly introduce you to the glycogen storage diseases. They are a group of genetic diseases due to defect in enzymes required for glycogen synthesis or degradation. So we've already studied synthesis and degradation. So the enzymes that were involved in glycogen synthesis and degradation, if there's any defect or deficiency of those enzymes, then it leads to glycogen storage diseases. They result either in formation of glycogen that has abnormal structure or in accumulation of excessive amounts of normal glycogen in specific tissues as a result of impaired degradation. So either the glycogen that is accumulating due to the impairment of the degradation, it can be normal glycogen, which is accumulating in excessive amounts, or it can be an abnormal form of the normal structure of glycogen. That is, it depends upon at what stage and which enzyme is deficient. So if it's like the starting enzyme is deficient, then the glycogen that will be accumulating will be normal glycogen. If some enzyme down the pathway is deficient, then an abnormal form of the glycogen will be accumulating. We'll see what it means. A particular enzyme may be defective in a single tissue such as liver, leading to hypoglycemia, or muscle, leading to muscle weakness, or the defect may be more generalized. So either the deficiency can only affect a single organ like liver or muscle, or it can be systemic. That is affecting different multiple organs like liver, muscle, kidney, intestine, myocardium. And the severity of the glycogen storage disease, it ranges from fatal in infancy to mild disorders. So it shows a wide spectrum of severity. That is, it can be fatal and it can also go to mild manifestations only. So that was briefly its introduction. Now your glycogen storage diseases, they have been categorized into various types. So we'll take each type in detail one by one. So talking about type 1, your type 1 is again subcategorized into two, 1A and 1B. So we'll see what's the difference between the two. So your type 1 glycogen disorder is also known as von Gerke's disease, and this is the most common glycogen storage disorder, and it is also the most asked. Okay, so it is important from both the aspects. The enzyme deficiency here is glucose 6 phosphatase. Okay, glucose 6 phosphatase. I hope you remember what was glucose 6 phosphatase. Your glucose 6 phosphate was changing into glucose. This was happening only in liver because the muscles already don't have this enzyme, and the enzyme was glucose 6 phosphatase. Okay, so your glucose 6 phosphate changes into glucose via the enzyme glucose 6 phosphatase. We have already studied this in glycogenolysis. I hope you remember. Okay, now this enzyme here is not working. This. So that means your glucose is not being formed and your glucose 6 phosphate is accumulating in the cell. So glucose 6 phosphate starts accumulating, and it is very obvious why will your glucose 6 phosphate start accumulating. Firstly, because obviously this enzyme is deficient, and also because now this glucose 6 phosphate cannot leave the cell. It will start accumulating inside the cell because we know that phosphorylated sugars do not have transporters for them in the plasma membrane, and it is too polar to cross the plasma membrane on itself. Now, this glucose 6 phosphate is an allosteric inhibitor of glycogenolysis. We have studied this as well in the regulation chapter. You see, all the chapters are very much integrated. So, you need to have the concepts very clear. What is activating, what is inhibiting. So, you need to memorize these factors. So, glucose 6 phosphate is an allosteric inhibitor of glycogenolysis, and it was an activator of glycogen synthesis. So it inhibits glycogenolysis. Now your glycogenolysis is being inhibited. That is, your degradation of glycogen is being inhibited. So your glycogen will start accumulating, obviously, because it is not being degraded anymore. So glycogen starts accumulating, but this glycogen is normal glycogen. Okay. So the glycogen starts accumulating, but this glycogen which is accumulating is not an abnormal glycogen, it is normal glycogen. Glycogen gets deposited in liver, renal tubule cells, leading to hepatomegaly and renomegaly. So excessive amounts of glycogen is being now accumulated because glycogenolysis has been inhibited. Now this leads to enlargement of the liver, which is known as hepatomegaly, and it is also being accumulated in renal tubule cells, leading to what is known as renomegaly. Other clinical features include hypoglycemia, ketosis, lactic acidosis, hyperuricemia, and hyperlipidemia. So instead of just mugging these clinical manifestations, I'm actually going to tell you the concept behind them. If you start relating the concept that this enzyme is deficient, so this pathway will not be working, so this will lead to accumulation of this. So, you know, the cascade of events that will form, you don't have to memorize or mug them up. You'll be able to relate them. So you just have to know, okay, this enzyme here is deficient, so this will not work, so this will increase, so this will rise, and everything. You will see what I'm talking, huh? So it says that the biochemical hallmarks of one Gurkis. We'll take one, each of them one by one, okay? So the first is hypoglycemia. Now, which enzyme is deficient? Your glucose 6 phosphate is phosphatase is deficient. So your glucose 6 phosphate, it changes into glucose, and the enzyme was glucose 6 phosphatase. Now, this enzyme here is not there. So your glucose 6 phosphate is accumulating, and your glucose is not formed. So when your glucose is not formed, what will happen? Hypoglycemia. Very simple. Because glucose is not being formed. And this is an example of classically fasting hypoglycemia. Now, why fasting? Because we already know that your glycogenolysis was working in the fasting state. So that is why this hypoglycemia is manifested in the fasting state. Now the other feature we're talking about is lactic acidosis. This was the first one. Okay. Now let's take the other example of lactic acidosis. Now again, so glucose 6 phosphate is accumulating and it is not converting into glucose. So what happens is your glucose 6 phosphate, now what fate it has? It enters anaerobic glycolysis. Okay. And we already know that anaerobic glycolysis, the product is lactate. So lactate starts increasing, leading to what is known as lactic acidosis. Very simple. Now, what do we have? The next thing. Okay, hyperlipidemia. Let's see how is hyperlipidemia occurring. Now your glucose 6 phosphate, as it was entering anaerobic glycolysis, it can also enter aerobic glycolysis. Okay. So when it enters aerobic glycolysis, what will it form? Pyruvate. And this pyruvate, it will ultimately form acetyl-CoA. Glucose 6 phosphate forms pyruvate. This pyruvate forms acetyl-CoA. Now your acetyl-CoA is increasing. Now this acetyl-CoA will enter fatty acid synthesis. Okay, so your fatty acids are being formed. Now your fatty acid, it combines with glycerol to form tags. Okay, so there is increase in hypertriglyceridemia and hyperlipidemia. Okay, so these are three. What is else is left? Okay, ketosis is left. How is ketosis occurring now? So we have seen that the early fasting state where glycogenolysis was taking place, that has been bypassed because your glycogenolysis is being inhibited. So you're immediately after the, see, there was early fasting state. We had studied this already, okay? 4 to 16 hours. And this is where your glycogen glycogenolysis was taking place. Okay, now what happens? Your glycogenolysis is not taking place. It does not happen. So you immediately enter into the second stage of fasting. That is from 14 to 16 hours. You enter that stage. And what was happening in this stage? In this stage, gluconeogenesis was taking place. And gluconeogenesis, what does it need? It needs oxaloacetate. So your oxaloacetate is being depleted now. Now what happens? To provide the energy, but the body still needs energy. What will happen? Beta oxidation of fatty acid will happen and it will lead to formation of acetyl-CoA. Now this acetyl-CoA has two fates. It can either enter TCA or it can either enter into ketone body synthesis. But we know that TCA needs oxaloacetate and oxaloacetate is already over. It is already being depleted. So your acetyl-CoA will enter into ketone body synthesis, causing ketosis. Okay. So your ketone body synthesis will increase. So we've taken all the points. Okay. Any left? Okay, hyperuricemia is still left. Okay, your hyperuricemia we'll just study in HMP shunt right now, that your glucose 6 phosphate can also enter into HMP shunt, and HMP shunt, it forms pentoses. So these pentoses, they form purines. So when more purine is formed, obviously more purine will be degraded, and the degradation product of purine is uric acid. So this leads to hyperuricemia. So we've discussed all the important chemical hallmarks of von Gerke's. Okay. So this was about von Gerke type A. Now there's another type which is known as type 1B. Now the defect due to endoplasmic reticulum glucose 6 transporter. Okay. So this is the same. There in type 1A, glucose 6 phosphatase, the enzyme itself was deficient. Here, the transporter, which is taking glucose 6 phosphate into the endoplasmic reticulum. We have already studied that your glucose 6 phosphate is present in the endoplasmic reticulum, and there were transporters, and here the reaction was taking place, and then the glucose comes out. Please refer to the lecture, huh? We've already studied. So this transporter is not working, this is defective. So obviously it will have the same manifestations as type 1A. Also, there is additional neutropenia and recurrent bacterial infection. That is, there is abnormally low count of neutrophils, which obviously leads to increased recurrent bacterial infections. So that was about type 1. Moving forward to type 2, that is also known as complex disease. It comes under lysosomal storage disorder. If you recollect, I had told you mostly it was cytoplasmic cycle, but 1 to 2% was taking place in the lysosome. So here the enzyme deficiency is in lysosomal alpha 1,4 alpha 1,6 glucosidase. And there's accumulation of normal glycogen in lysosomes. There are two types of variant, juvenile and adult. So in the juvenile variant, death from heart failure occurs by the age of 2. Moving forward, we have the type 3 glycogen storage disease, also known as limit dextrinosis, Forbe or Cory's disease. The enzyme deficient here is D-branching enzyme. It again has two subcategories, type 3A and type 3B. When D-branching enzyme of both liver and muscle is defective. So in type 3A, the debranching enzyme of both liver and muscle is defective or deficient, and in the type B, only the debranching enzyme of liver is defective. Okay, glycogenolysis does occur, but partially, as glycogen phosphorylase is working, but debranching enzyme is not working. So if you remember glycogenolysis, what was happening was this was your glycogen. So glycogen phosphorylase was working initially, and it would remove glucose as glucose 6-phosphate units, and then your debranching enzyme was coming, which was transferring a trisaccharide unit. So now this debranching enzyme is not working, but your glycogen phosphorylase is still working. So that's why glycogenolysis is occurring partially, because your glycogen phosphorylase is working, but debranching enzyme is not working. And we already know that the end product of glycogen phosphorylase was limit dextrin, if you remember. So abnormal glycogen, which is limit dextrin, that is accumulating. So here in von Gerke's, the glycogen that was accumulating was normal glycogen, but here in Cory's disease, limit dextrin is accumulating. Hence the name limit dextrinosis. It also has another name which is known as limit dextrinosis. Again, there's fasting hypoglycemia, there's hepatomegaly. So it is a milder form of type 1. Moving forward, we have type 4 glycogen storage disease, which is also known as amylopectinosis or Anderson's disease. The enzyme deficient here is branching enzyme, accumulation of abnormal glycogen. So here the branching enzyme is deficient. So your branches are not being able to form. So what will you have? A polysaccharide with very few branches. The outer branches will be very long, because since branching is not taking place. Death from heart or liver failure before age 5. Okay, I'll give you a short mnemonic to remember this. A, A, B, C, D. What does this A, B, C, D means? A for Anderson. So in Anderson, your branching enzyme is defective. C for Cory's, and in Cory's, your debranching enzyme is defective. So this is just like simply to memorize. Okay. Type 5 glycogen storage disease, also known as McArdle syndrome or myophosphorylase deficiency. So as the name already suggests, the enzyme deficiency is in muscle glycogen phosphorylase, normoglycemia, very obvious because muscle glycogen was involved in providing ATP or energy to muscles during contraction or exercise. It was never involved in the first place to maintain blood glucose level. That was the function of liver glycogen. So obviously there will be no hypoglycemia, but there will be exercise intolerance. And other than that, the patient is normal and well developed. Exercise intolerance, I've just said, because there will no ATP or energy during exercise or muscle contraction. Okay. Type 6, also known as Hirsch disease. Here the enzyme deficiency is in hepatic glycogen phosphorylase. Okay, so there will be hepatomegaly and accumulation of glycogen in the liver. Type 7, also known as Torrey's disease, the enzyme deficiency is in muscle and erythrocyte phosphofructokinase 1. Now you might think that phosphofructokinase 1 is a glycolysis enzyme. It is a glycolytic enzyme. And why is it being mentioned here? Because if you remember, in muscle glycogenolysis, there was no glucose 6 phosphatase. So glucose 6 phosphate was actually entering glycolysis to provide ATP. Therefore, this is how your phosphofructokinase 1 becomes a part and is related to glycogenolysis. So, muscle glycogen is abnormally high. There's exercise intolerance and hemolytic anemia because RBC is solely dependent upon glucose as a fuel, whether it be in fed or fasting state. So, when there is deficiency in phosphofructose kinase, it will lead to hemolysis of the RBCs, lysis of the RBCs. Okay, so that was about your glycogen storage diseases. Mhm. Okay, moving forward, let's start with HMP pathway. Again, let's begin with the clinical scenario. Okay, here in this clinical scenario, which we had taken, now can you tell me what will be the answer here? It clearly says that your glucose 6 phosphatase is deficient. So what will be? Glucose 6 phosphatase we have already studied. Glucose 6 phosphatase is deficient in von Gerke's. So you know this is the answer. Okay. Okay, let's begin with HMP pathway. So taking a case scenario, a 45-year-old woman presents to the emergency room with complaints of back pain, weakness, and dark colored urine. On examination, she is found to have hemolytic anemia and jaundice. Laboratory tests reveal anemia, elevated bilirubin levels, and the presence of Hen's bodies in red blood cells. The patient has a history of exposure to certain drugs and chemicals. The healthcare team suspects oxidative damage to red blood cells leading to hemolysis. Which enzyme of HMP pathway is likely to be deficient in this patient, leading to oxidative damage in red blood cells? Again, this is a very simple question, although might seem a little lengthy, but let's study HMP pathway and then it will be easier for you to answer that. So your HMP pathway is also known as pentose phosphate pathway because the reactions involve the intermediate formation of pentose phosphatases. So as we study, you will see that the intermediates form, they are pentose phosphatases. So one of the names of HMP is also pentose phosphate pathway. It is also known as Dickens-Auricle pathway. It is also known as shunt pathway because the glucose, instead of entering glycolysis, is shunted to this pathway. So hence the name shunt pathway. It is also known as phosphogluconate oxidative pathway. Phosphogluconate is one of the intermediate, hence name on that. It is also known as hexose monophosphate. In glycolysis, we have seen a few bis intermediates being formed, like fructose 1,6 bisphosphatase, 1,3 bisphosphoglycerate. But here you will notice that only monophosphates are formed, therefore also known as hexose monophosphate pathway. The names are important because sometimes if you know HMP only by the name of HMP pathway and you do not know that it is also called shunt pathway, and in the exam it's like write a short note on shunt pathway and you'll be like, what is shunt pathway? So it is like you should know the names, all the names of a particular cycle, all the different names of the same cycle. Okay, so what is the significance or the importance of HMP pathway? That is generation of NADPH and generation of pentoses. Pentoses are important because your pentose sugar forms the part of nucleotide, and nucleotides are the building blocks of your DNA. So important for DNA and RNA synthesis. ATP is neither utilized nor produced by HMP pathway. So energy production is not the goal of HMP pathway. HMP's pathway's significance is to provide NADPH and to provide pentoses, and it is a cytoplasmic pathway. Now your HMP pathway has two phases: oxidative phase, which is irreversible, and non-oxidative phase, which is reversible. Oxidative phase. One molecule of glucose 6-phosphatase is oxidized to generate two molecules of NADPH, two molecules of NADPH it says, and one molecule of ribulose 5-phosphate with liberation of one molecule of carbon dioxide. Okay, let's see what is it saying. So see glucose 6-phosphate here, two molecules of NADPH. So one molecule here and one molecule here. So one glucose 6-phosphate is oxidized to give two molecules of NADPH with the liberation of one molecule of carbon dioxide along with the formation of one molecule of ribulose 5-phosphate. Let's see how. Your glucose 6-phosphate, this is your glucose 6-phosphate. It is a 6-carbon compound which is acted upon by glucose 6-phosphate dehydrogenase. Here, it is acted upon by glucose 6-phosphate dehydrogenase, which is a rate limiting enzyme. Okay, so your glucose 6-phosphate dehydrogenase, which is a rate limiting enzyme, it converts this glucose 6-phosphate into 6-phosphogluconolactone, which is then acted upon by hydrolase. And what is formed? 6-phosphogluconic acid is formed. Now you see, all these are 6-carbon compounds. Okay. Now this is acted upon by 6-phosphogluconate dehydrogenase. Okay. Now dehydrogenase. So what is the accepting molecule here? NADP plus. So NADP plus accepts that hydrogen which is removed from here from 6-phosphogluconic acid, and it forms NADPH, the second molecule of NADPH. The first molecule was formed in this reaction. The second molecule of NADPH is formed, and you see this is a 6-carbon compound, but your ribulose 5-phosphate, it is a 5-carbon compound. So one carbon dioxide was released during this, and this carbon dioxide is coming from this COH group. So your ribulose 5-phosphate is formed, and this is irreversible. So this is the first phase of HMP, which is known as the oxidative phase. It is irreversible. What happens in this is that your one molecule of glucose 6-phosphate oxidizes to give two molecules of NADPH and one molecule of ribulose 5-phosphate with the liberation of one molecule of carbon dioxide. And the rate limiting enzyme of this phase is glucose 6-phosphate dehydrogenase. Okay. Now moving on to the next phase, that is the non-oxidative phase of HMP pathway. This includes reversible reactions. Okay. These reversible reactions, they permit the ribulose 5-phosphate, which was produced by the oxidative phase, to be converted either to ribose 5-phosphate needed for nucleotide synthesis or to intermediates of glycolysis, that is fructose 6-phosphate and glyceraldehyde 3-phosphate. Let's see how. I hope the diagram is clear. Can you see it? Okay. Is that fine now? Okay. So this was your first phase, oxidative phase, and in the oxidative phase, we have ribulose 5-phosphate being formed. We've just seen these reactions. So you take 3 ribulose 5-phosphate, okay? That means you have oxidized 3 molecules of glucose 6-phosphate to get 5 ribulose 5-phosphates, 3 molecules. Now what happens is your ribulose 5-phosphate, by the action of enzyme epimerase, it changes into xylulose 5-phosphate. Your ribulose 5-phosphate is a 5-carbon keto sugar. Okay, and your xylulose 5-phosphate is also a 5-carbon keto sugar. So both are 5-carbon keto sugar. What is the difference? The difference lies in carbon 3 in the orientation of hydroxyl and hydrogen ions. So these are epimers. In isomerism, you must have studied that ribulose 5-phosphate and xylulose 5-phosphate, they are epimers of each other, although both are 5-carbon keto sugar. But at carbon 3, due to difference in orientation of hydroxyl and hydrogen ions, they are epimers of each other. So your ribulose 5-phosphate epimerizes to xylulose 5-phosphate and the enzyme is 3-epimerase. Okay. This is the same reaction. So 2 ribulose 5-phosphate are changing into xylulose 5-phosphate and 1 ribulose 5-phosphate changes into ribose 5-phosphate. Now, ribulose is a 5-carbon keto and ribose is an aldehyde. So this is ketoisomerase. Enzyme is ketoisomerase. Okay. Now what you get? Xylulose 5-phosphate, ribose 5-phosphate, and xylulose 5-phosphate. Now this xylulose 5-phosphate and this ribose 5-phosphate, they are acted upon by trans-ketolase. So this trans-ketolase, it transfers 2 carbon atoms from this xylulose 5-phosphate, which was 5 carbon atom, to this ribose 5-phosphate to form 7 carbon atom pseudoheptalose, and itself it forms glyceraldehyde 3-phosphate. Now this glyceraldehyde 3-phosphate and pseudoheptalase, they are acted upon by transaldolase to form fructose 6-phosphate and erythrose 4-phosphate. Erythrose is 4 carbon and fructose is 6 carbon. So 3 carbon from pseudoheptalose are transferred to glyceraldehyde 3-phosphate to form 6 carbon fructose 6-phosphate, and here 4 carbon are left. Itself it forms erythrose 4-phosphate. Now this erythrose 4-phosphate, it combines with that third xylulose 5-phosphate, second xylulose 5-phosphate, which was formed from this third ribulose 5-phosphate. Okay, it combines with that and is acted upon by trans-ketolase to form fructose 6-phosphate and glyceraldehyde 3-phosphate. So you see, these are all intermediates of glycolysis now. So ultimately you get from this fructose 6-phosphate and fructose 6-phosphate, you get glucose 6-phosphate, here glucose 6-phosphate, and here half glucose 6-phosphate because glyceraldehyde is 3 carbon. So you get half glucose 6-phosphate. So this was all about the non-oxidative phase, which was reversible. So all non-oxidative phase reactions are reversible, so they can give rise to pentoses here. See, this was your ribose 5-phosphate formed here. Now this ribose 5-phosphate goes into synthesis of nucleotides, RNA, DNA synthesis. So all these reactions of the non-oxidative phase, they are all reversible. So they can be reversed to give this ribose 5-phosphate in cells when needed for the synthesis of RNA, DNA, especially like in the skin and all where there's more wear and tear, so more synthesis of DNA is needed. Okay. Now, organs where your HMP operates. The irreversible oxidative phase is particularly important in those organs in which there is active NADPH dependent biosynthesis of fatty acids or steroid hormones. So, what are those organs in which there is reductive biosynthesis of fatty acid taking place? It is liver, lactating mammary gland, and adipose tissue. And biosynthesis of steroid hormones take place in testes, ovaries, placenta, adrenal cortex. And in RBCs, which need NADPH to keep glutathione in reduced state. So your irreversible oxidative phase is particularly taking place in all these organs where there is reductive biosynthesis of cholesterol, fatty acid, and steroid hormones going on. And your reversible non-oxidative phase occurs in almost all tissues to produce pentoses because DNA and RNA synthesis is taking place in almost all the organs. So your reversible non-oxidative phase takes place in almost all tissues to form pentoses. Now regulation, glucose 6-phosphate dehydrogenase, as I mentioned, is the rate limiting enzyme. It was involved in the first step of oxidative phase and it is inhibited by NADPH. So high levels of NADPH will inhibit glucose 6-phosphate dehydrogenase and low levels of NADPH will enhances its activity. Insulin upregulates upregulates the expression of the gene of glucose 6-phosphate dehydrogenase. So that is in the well-fed skin when there's insulin, the activity of the gene of glucose 6-phosphate dehydrogenase will be increased, therefore increasing the overall HMP pathway. The non-oxidative phase is controlled by the requirement of pentoses. Okay, so those sites where there's requirement of more pentoses, like there's more DNA synthesis taking place, then your non-oxidative phase will be acting more. Now coming on to uses of NADPH. NADPH is required for the reductive biosynthesis of fatty acids, steroids, and cholesterol. We had just seen what were the organs where the synthesis is taking place. Now your NADPH, do not confuse this NADPH with NAD. Okay? Your NADPH is different from NAD in having an additional phosphate group. Okay? Your NADPH is considered as a high energy compound, just like NADH, but the electrons of NADPH they are used in reductive biosynthesis. That is, in the synthesis of these fatty acids and steroids and cholesterol, the reactions that require electrons, so for them, the electron donor is your NADPH. Okay, unlike NADH, where it was going into the electron transport chain to provide ATP, your NADPH is not going into an ETC to provide energy. Instead, it is involved in reductive biosynthesis. And in those reactions where there is requirement of electron, your electron donor is NADPH. So this is one use of NADPH. Now, free radicals scavenging. Okay, so your free radicals are continuously being formed in the cells, which can damage the cells. Your free radicals are highly reactive and it can damage the cells. The partial reduction of molecular oxygen leads to the formation of hydrogen peroxide. It leads to the formation of superoxide. All these are highly reactive species, reactive oxygen species known as ROS, and they can be really damaging for the cell. So it is very important for the body to get rid of these ROS or free radicals. So they scavenge them. The body has this protective mechanism, which is known as free radical scavenging. And this free radical scavenging is dependent on glutathione in reduced state. Okay, so to keep glutathione In reduced state, NADPH is needed. See, this was your glutathione. This is your glutathione. This is here in reduced state. Now, this glutathione is able to detoxify this hydrogen peroxide to water in the presence of the enzyme glutathione peroxidase. Okay, so your free radical has been scavenged. This ROS has been detoxified to water. But at what cost? That this reduced glutathione changes into oxidized glutathione. So this free radical scavenging can take place only when this glutathione is in reduced state. So you need to maintain this glutathione in reduced state. So for that, what do you need? You need glutathione reductase enzyme, and the reducing equivalent here is NADPH. So NADPH will change into NAD plus and it will give you reduced glutathione. So your reducing equivalent here is NADPH, which helps in maintenance of glutathione in reduced state. So free radicals are continuously being produced in the cells, which can damage the cell. Free radical scavenging is dependent upon glutathione in reduced state. To keep the glutathione in reduced state, NADPH is needed. Now, organs where free radical scavenging is important, it is important in RBC to maintain the membrane integrity. It is important in lens to maintain the transparency of lens. If free radicals start accumulating, the lens will turn opaque. Now, in hemoglobin, normally in hemoglobin, iron is in ferrous state, that is Fe2 plus. And if iron becomes ferric, that is Fe3 plus, hemoglobin is known as methemoglobin. So one valency is lost, so it cannot carry oxygen. So free radical scavenging keeps iron in ferrous state. So it becomes very important to keep the iron of hemoglobin in ferrous state. The microsomal cytochrome P450 system in the liver brings about detoxification of drugs and foreign compounds by hydroxylation reactions involving NADPH. Now, to explain this, you will have to know a little about cytochrome P450. You will study about the enzymes classification in detail in enzymology, but here just know that your enzymes are classified into 7 different classes. 1, 2, 3, 4, 7. So out of those 7, like oxidoreductases, transferases, and etc. Like this, you have 7 classes. Okay. So one of those classes, that is oxidoreductases, it has again subclasses, 6 subclasses. 1, 2, 3, 6 subclasses, and one of those subclass is your oxygenase. Now, what are oxygenase? Oxygenase are those enzymes which directly add oxygen to the substrate. Now, these oxygenases, they can be again classified into 2 types: monooxygenase and dioxygenase. Monooxygenase are those which add one atom of oxygen, molecular oxygen, to the substrate. Okay, one atom. And dioxygenases, they add both the atoms of oxygen. So your cytochrome P450 is an example of monooxygenase. So your cytochrome P450 is an enzyme, monooxygenase enzyme. It is a heme containing enzyme. Okay. Now when it says, so you know what is cytochrome 450 system. Okay. So your microsomal cytochrome 450 system in the river, it brings about detoxification of drugs. So this enzyme is involved in detoxification of drugs. You will study this in great detail in xenobiotics. Anything that I'm explaining here is not extra. It's just that you will study all of this in different chapters. But for understanding at the moment, I'm explaining you things here as well. So you know what is cytochrome 450? So it is involved in detoxification of drugs. And during this detoxification, it undergoes hydroxylation reactions. And those hydroxylation reactions are requiring NADPH. Okay. Now, macrophage bactericidal activity. NADPH is required for the production of ROS by macrophages to kill bacteria. See here. What happens is, like, your phagocytic cells, like your dendritic cells, these are your professional phagocytes, like macrophages, neutrophils, dendritic cells. So these phagocytic cells, for example, this is one of your phagocytes. They have receptors, and this is some bacteria. Okay. So this bacteria is engulfed, phagocytosed by this phagocytic cell. It enters here inside and it forms what is known as phagosome. Okay. Now this phagosome, it combines with the lysosome to form what is known as phagolysosome. Okay. So this is your see, this is what is happening. This was your bacteria. Bacteria, it is attacked by two antibodies, and then the receptors for those antibodies is present on the phagocytic cell, and then they are engulfed, phagocytosed. Ingestion takes place of the microorganism here. Then the vacuole that forms around the engulfed substance, that is the bacteria here, is known as phagosome. Now this phagosome combines with lysosome to form what is known as phagolysosome. Now in the membrane, what is present? NADPH oxidase is present. This enzyme is present. So it converts the molecular oxygen that is present in the nearby area into superoxide. Now this superoxide is acted upon myeloperoxidase and combines with chloride to form hypochlorous acid. Now this hypochlorous acid has bactericidal function and it kills the bacteria. See, I'll repeat this again. To macrophage bactericidal activity, NADPH is required for the production of ROS macrophages to kill bacteria. See, this superoxide which was formed here, this is your ROS. That is reactive oxygen species. How was it involved in killing the bacteria? The bacteria was through this cascade of events phagocytosed, and then the enzyme NADPH oxidase, which was present here. This requires for its functioning NADPH. So this is how your NADPH is involved to convert this molecular oxygen into reactive oxygen species, which has bactericidal function on its own, and it also has bactericidal function by forming hypochlorous acid. So if your myeloperoxidase is deficient, you'll not be as prone and susceptible to infections. But if your NADPH oxidase is deficient, then you are more susceptible to infections. Okay. Now moving on to clinical correlations. Glucose 6-phosphate dehydrogenase deficiency. It is an X-linked inherited disease. It is the most common disease-producing enzyme abnormality in humans. Decreased glucose 6-phosphate dehydrogenase will lead to decreased NADPH. Obviously, when the enzyme is not working, your glucose 6-phosphatase phosphate dehydrogenase is deficient, then your HMP pathway will not work, and then there will be decreased NADPH. Decreased NADPH means decreased free radical scavenging and increased free radicals. Your free radicals will not be scavenged. So this will lead to accumulation or an increase in free radicals. And increased free radical means more damage. And there will be drug-induced hemolytic anemia. Now your NADPH were required for free radical scavenging, as we have seen that your NADPH was maintaining glutathione and reduced it. Your NADPH also helps to maintain other proteins in their reduced form. In the absence of this scavenging, your proteins, membrane proteins, they will be oxidized. And these oxidized proteins, they lead to formation of denatured proteins that form insoluble masses called as hence bodies. And these hence bodies are attached to the RBC membrane. Now, what is drug-induced hemolytic anemia? There are certain drugs like sulfa drugs and anti-malarial drugs like primaquine. What they do is they lead to peroxide formation. So as it is, NADPH was decreased and there was decreased free radical scavenging, but and formation of more free radicals will lead to lysis of the RBCs. Therefore, this is known as drug-induced hemolytic anemia. And if you consume fava beans, they have the same effect. So glucose 6-phosphate dehydrogenase deficiency due to consumption of fava beans is known as favism. Now, Wernicke's Korsakoff syndrome, it is due to thiamine deficiency. If you see this chart again here, in this step we needed trans ketolase. Now this trans ketolase here, this requires TPP. TPP, thymine pyrophosphate as a coenzyme. So this trans ketolase, it requires thymine pyrophosphate as a coenzyme, TPP. This is the active form of thymine. So this enzyme helps you to know the thymine status of the cell, okay? So in case of thymine deficiency, your trans ketolase will not be functional, which will lead to this syndrome, Wernicke's Korsakoff syndrome, okay? So that was all about your HMP shunt. Let's do a few questions. An infant has hepatomegaly, hypoglycemia, hypolipidemia, acidosis, normal structured glycogen deposition in the liver. What is the diagnosis? So in case anyone of you has any doubt, you can send in your queries. And also I can see that not everyone is answering the poll questions. Our next class will be on GAGs, that is glycosaminoglycans, which will be on 28th February. So with that, we come to the end of this session. If you have any doubts or queries, you can send them in. You can also contact to us later on with your queries via email